Good morning to everybody. Today we are going to discuss about the Apomixis. We have the main chapter in the embryology for the BSc second year students, botany. This is Dr. DNR, Assistant Professor of Botany, Government Degree College, Warangal, Telangana State. Is the main process Apomixis. Usually, in all the plants, Amphimixis is happening. That means meiosis and reduction division, fertilization, meiosis or reduction division and fertilization process will happen. By that, it forms the diploid zygote. It forms the seed and it germinates and it forms the yolk. Right. But in some of the plants, it lacks the meiosis and fertilization. Though it lacks the meiosis and fertilization, it will form the diploid seeds. As usual, general Amphimixis plants. That is called Apomixis. Okay, Apomixis is first discovered by Leuwenhock and word given by Winkler. Okay. Here, Apomixis process, usually this yellow one is the general process Amphimixis. Here, meiosis, double fertilization is happening there. But in Apospory, it lacks the meiosis and also it lacks the fertilization. Though it lacks the meiosis and fertilization, it forms the new seeds, germinate, and it as usual like a Amphimixis plant, general plants. These processes are locked, that is Amphimixis. The blue, red, green, these three comes under the Apomixis. Yellow one is Amphimixis. Okay, that means general fertilization that is happening in all the angiosperms commonly. But the Apomixis is specific, it is happening with the special features that though it is not happening general meiosis and reproduction, fertilization as usual characters of the reproduction, it is forming the seeds as like other plants. Okay. Here, Apomixis derived from the Greek word. I will explain you. Apo means away from the mix, and mixed means mixing, mingling. Reproduction involving meiosis and fertilization in flowering plant is called Amphimixis. Reproduction does not involve union of male and female gametes, that means fusion, and meiosis, fertilization. These three are locked is called Apomixis. There is no fusion of the gamete, there is no meiosis happening, reduction is happening, no double fertilization is happening. But the seeds are formed, that is Apomixis. It was first discovered by Leuwenhock in 1799 in citrus seeds. Okay, and the term Apomixis was introduced by Winkler in the year 1908. Okay, it's defined as the substitution of the usual sexual system, Apomixis, by the form of reproduction which does not involve meiosis and syngamy. Okay, Apomixis is widely distributed among the higher plants. More than 300 species belong to the 35 families. 35 families of 300 species is showing the Apomixis. It is mostly common in Graminae, Poaceae, and Compositae, Asteraceae, Rosaceae, and Rutaceae. Among the major cereals, maize, wheat, and pearl millets have an apomictic relationships in the monocots. Okay. Here you can find out these are the developmental pathway of various types of the Apomixis. Diplospory, pale urine reduction division, Apospory, within the embryo sac, other cells are found the diploid spores, and adventive embryos, somatic cells from the embryo other than embryo sac. These are the three various types which are present in the Apomixis. Okay. Here, the Maheswari classifies the Apomixis into two types. That is, recurrent Apomixis and non-recurrent Apomixis. Recurrent Apomixis means in this type, the embryo sac develops in the diploid egg mother cell or from some other diploid cells of the embryo sac without the fertilization. It is forming from the diploid cell without the fertilization. Then what happens? The egg has the normal diploid number of chromosomes, the same as in the mother plant. Okay, the embryo subsequently develops from egg nucleus without the fertilization. So it is coming from the egg nucleus without the fertilization. It is happening. It consists of two types: vegetative propagation and agamospory. Agamospory can be divided into Adventive embryony, Diplospory, and Apospory. Okay. Here, what happening here is diploid egg mother cell or from the diploid cell of the embryo sac. Without the fertilization, it is happening. That's what the recurrent Apomixis. Okay. Then in the recurrent Apomixis type, first one is vegetative reproduction. Usually, vegetative propagule plant which can which can do the reproduction by the vegetative propagules. This is the common type in the vegetative propagule plants. Okay, plants propagate by the any part other than the seeds. New plants develop from the part other than the seed. Okay, that is vegetative reproduction. Example of bulbs. Intricularia imperialensis or bulbs. Allium, Cephalium, runners, Lanta, Arvensis, Pudina, and suckers, Chrysanthemum, Philippomucca. And second one is Agamospory. This is the process by which embryo are formed by eliminating meiosis and syngamy. By eliminating meiosis and syngamy, it forms the seeds. These are two different types of the Agamospory recognized based on whether embryo developed from the cells of the unreproduced female gamete. If it from the unreproduced female gamete, it is a gametophyte. Or directly from the diploid sporophytic cell of the ovule, such as nucellus or integument. This is sporophytic part of the ovule. That's why this is sporophytic or Adventive embryony. Here, like gametophytic and sporophytic embryony, both the type of embryony will happen in the Agamospory. Okay. And in that, first one is Diplospory. Diplospory, what happens, how it forms? The unreproduced embryo sac is derived from the megaspore mother cell, either directly by mitotic division or by aborted meiotic division. By stopping the meiotic division or by directly it goes to the mitotic divisions. It forms the diploid of the sac embryo. These three major types of the Diplospory are been reported, named after the plant, in which they occur. They are Caraxagam type, Edgeris type, Antenaria type, and Allium type. We will go through each single one of the Diplospory. Here, Diplospory means we understood. It is developing from the diploid cells, megaspore mother cell MMC. Directly it is going the mitotic division. There is no meiotic division, so it is diploid. Diploid cell is forming the zygote seed. So this is Diplospory. Okay. Here we can see in the Caraxagam type, meiotic phase is initiated, but then the process is aborted. Result, what happened to unreduced dias? To here, unreduced dias, one of which gives rise to the embryo sac. One of which, the uh, micropylorium, it takes the divisions, mitotic divisions, not the meiotic division. Take the mitotic division by the way from the core cells and from the egg cell and from the embryo sac, as like polygonum type. Okay, example is Taraxacum or Winkler. And second one is Agamospory. Here, meiotic division is arrested. Mitotic division is takes 3 times to take the mitotic divisions. Okay, 2 more mitotic divisions of the nucleus. Give rise to the 8 nucleate. How many? 8. 8 nucleate embryos are follow the equational division following the meiotic propagules. The starting the meiotic propagules to take the further mitotic division, not the meiotic division. Meiotic division aborted and mitotic division is started. Taken the more divisions by the from the egg cell embryo sac. Taraxacum and Agamospory are known as a meiotic diplospory because they involve modification of meiosis. Here is a modification, the pre-meiosis. Meiotic prophase is there, but it's not fully meiotic stage. The reinstitution of the nucleus is happened here. Okay. Here, egg cell embryo sac are formed. Example, Exeris DNR. Third one is attenuated type. Here there is no meiotic space, meiosis. Unreduced megaspore, the long interface, and it acts as a functional megaspore. After the long interface, it acts as a functional megaspore through the mitotic division. It is not entering the meiosis, it is acting as an unreduced megaspore, it is taking rest. After long interface, it is acting as a functional megaspore. In the interface, in between the mitotic division, interface, it is modifying and it acting as a functional megaspore for mitotic divisions. Then what happened? 2 cells, 4 cells, 8 cells. It form the 8 cells, embryo sac, like polygonum. It shows wide taxonomic distribution. Most of the species having this type of diplospory type, that is attenuated type. Okay, example in Apospory, Propagules. And fourth one is alien type. This alien sepal. Okay, Liliaceae family. The pre-meiotic chromosome doubling is the cause of unreduced embryo sac formation. This is the main feature. Pre-meiotic chromosome doubling is the cause of unreduced embryo sac formation. Chromosome number double by endomitosis. Okay, here chromosome number is double by endomitosis, and the ensuing meiosis results in a dyad of unreduced cells, not the reduced cells, unreduced cells. 2 subsequent mitosis in chelagal dyad cells resulting in 8 nucleate embryo sac. Then what happens whenever the unreduced dyads are formed, then subsequently the chelagal dyad cells, it goes the subsequent mitotic divisions, then it forms the 8 nucleate embryo sac, that is helium mutants. And next one is Apospory. We studied up to this is Diplospory. Okay, under the gametophyte Apomixis. Okay, second one is Apospory. It is the most common mechanism of plants. First reported by Rosenberg in 1907. Megaspore mother cell undergoes the normal apospores are formed, but gradually disappear. A new cellular cell becomes activated and develops into a diploid embryo sac. This type of apospory is called somatic apospory. Why? Because here new cellular cells, this is a sporophytic cell, is somatic cell activate. That's why it is a somatic apospory. It is somatic apospory. Example, Heracium and Poppy. They are similar in appearance to the meiotic megasporophytes and they differentiate close to the meiotic megasporophyte and develop into an Apomixis embryo sac. Once the aposporous initial cells differentiate, they immediately enter into meiosis to produce the embryo sac. Some ovules can contain multiple embryo sacs, and depending on the species, the structure of the embryo sac may be quite different from that seen in the sexual process. Okay. The initiation of the apospory embryo sac can occur together with the sexual one, or it can displace or inhibit sexual embryo sac formation. It initiates with the gender sexual reproduction, but it what happens? It displays or inhibits the sexual embryo sac formation. Then asexual embryo sac will form by the apospory. Here also two types of that, Heracium type and Formican type. Heracium type, a new cellular cell at the cheladal end. New cellular cell at the cheladal end of the tetrad becomes activated and start the developing into the aposporus. Unreduced embryo sac. All the 4 megaspores gradually degenerate and the aposporic embryo sac. Here what happens? All the 4 megaspores gradually degenerating and aposporic embryo sac mature. Aposporic embryo sac mature from the 8 nucleus. It is replacing the megaspore mother cell. Then the Amphimixis embryo sac is replaced by the Apomixis embryo sac. Example is Heracium flagellate. And next one in the apospory is the panicum type. In this aposporous embryo sac, it's 4 nuclei, not the 8 nuclei, 4 nuclei organized into 3 cells, exoperators and the 1 polar nucleus. Antipolar cells are absent. Okay, like cypress. In cypress also 4 cell embryo sac is present. Okay, the Poya, Arctica, Panicum and Penicillin. The examples. Okay. Next one is Adventive embryony. This is the sporophytic apomixis. First two, Diplospory and Apospory are gametophytic apomixis, and third one, Adventive embryony, comes under the recurrent apomixis, is Adventive embryo. An embryo arises directly from the diploid sporophytic cell, either from the nucellus or from the integument. It is also called as sporophytic budding because here gametophytic phase is completely absent. Gametophytic stage is completely absent. Only sporophytic phase in the ovule, somatic cells, nucellus or integument will form the embryo. That means zygote. It is close to the vegetative propagation, but differ from it essentially in 2 ways. One is it has retained the seed habit, like as usual Amphimixis. 1 or more diploid sporophytic cells undergo the stages of embryogeny and grow into mature embryos. Inside the normally developed sexual embryo sacs, 1 or 2 other Apomixis embryo sacs also will be enter into the embryo sac. zygote embryo either degenerate or compete with the apomixis embryo. It usually leads to the formation of more than one embryo in seeds. It is common in our citrus, mangifera. We already studied in the polyembryony. Whatever the new cellulose embryos that are penetrating into the embryo sac, occupying the space of embryo sac. By that, what happens? Here, zygotic embryo may degenerate or compete with the apomixis. Okay, and also in the Buxaceae, Coctaceae, Propagules, and Myctaceae, Archidaceae family. Here we already studied in the polyembryony. You can see Adventive embryos which are formed from the new cellulose. Here you can see from the new cellulose, Apomixis embryos are formed. Somatic embryos are formed. Okay, and then non-recurrent Apomixis, second one. As per Panchana Maheswari, first one is recurrent Apomixis. Second one is non-recurrent Apomixis. The recurrent Apomixis, we studied the vegetative propagation, Diplospory, Apospory, and Adventive embryo. The non-recurrent Apomixis is usually started. This upward embryo sac develops after the meiosis. Develops into an embryo without the fertilization. There the diploid embryos are formed. Here in the non-recurrent Apomixis, haploid embryos are developed. Okay, without the fertilization. These type of Apomixis occur very rarely and is primarily of genetic interest. Okay. The plant produces a haploid and sterile. It is common in Solanum nigrum and Leuwenhock species. It consists two types, haploid parthenogenesis and haploid haploidy. We will discuss about these two. First one is haploid parthenogenesis. Here in this, the embryo develops directly from the haploid egg cells. Development of embryo from egg without the fertilization. Usually, it fuse with the male gamete and it forms a zygote. But here, in the haploid parthenogenesis, development of embryo from the egg without fertilization. Two types. Haploid parthenogenesis and diploid parthenogenesis. Haploid parthenogenesis occur in Solanum nigrum and diploid parthenogenesis occur in Heroxypum. Haploid apogamy. Here, development of embryo from other haploid cells inside the embryo sac, other than the egg cell. If it is generated from the egg cell, that is haploid parthenogenesis. If it is other than the egg cell without fertilization, and as a result, the embryo developed is also haploid. Okay, it is forming from the egg cell, that is haploid parthenogenesis. If it is forming from the other cells, other than the egg cells of the embryo sac, without fertilization, is called haploid apogamy. The embryo developed is also haploid. And other non-recurrent Apomixis are androgenesis. Haploid embryo developed from the haploid sperm nucleus. Apogamy, development of sporobite directly from the gametophyte without the fusion of the condoms. Such sporobite have the same chromosome number as the gametophyte from which they have been derived. Okay. Usually this is happening in the pedophytes. So now, parthenogenesis. Haploid embryo developed from the haploid egg. That is parthenogenesis. Okay, without the fertilization. Pseudogamy. Pollination serves as a stimulus for embryo development. egg and sperm nuclei do not fuse. Fusion of polar nuclei with one of the sperm nuclei may occur to produce endosperm. Pollination serves as a stimulus of embryo development. But again, sperm nuclei does not fuse. Fusion of polar nuclei with one of the sperm nuclei may occur to produce endosperm. Syngamy. Haploid sperm nucleus enters the egg but does not fuse to the haploid egg nucleus. Each nucleus divides independently, creating haploid embryo that contains sections of male and female organ. Syngamy. Okay. The haploid sperm nucleus entering into the egg, but it is not fusing. Separately, excellent sperm cell, it is forming the haploid embryo with the two sectors. Okay, these are the another non-recurrent apomixis. And in apomictic species, sexual reproduction is either suppressed or absent. When the sexual reproduction also occurs, the apomixis is termed as facultative apomixis. Reproduction in plant by only apomixis when the sexual reproduction is absent is called as obligate apomixis. Apomixis with the sexual reproduction is called facultative apomixis. Apomixis without the sexual reproduction is called obligate apomixis. Many crop species shows apomixis, but it is generally facultative apomixis. But usually, obligate apomixis may not be possible without the stimulus of fertilization, fusion. It may not form the seeds. And what is genetics behind the apomixis? Gametophytic apomixis concern only the female side. Why? These are generating directly from the female gametophyte, megaspore mother cell only. So we have the all characters of mother plant. Nogler 1944 and Khrushchev 1942 noted that development of gametophyte apomix is affected either by dominant gene or linked genes formed on the chromosome. In most of the plants, obligate apomix are formed. Okay? The main reason, genetic reason is dominant gene or linked gene. And next one is early expression of the super gene leads to the apospory, while its late expression is called the diplospory. And many crop species show apomixis, but it is generally facultative. But obligate apomixis does not occur in any plant. Why? Along with the sexual reproduction only, it will be stimulated to form the embryo and seed. Without the stimulus, they can't produce the seed. Okay, Murthy opined out products of apomix may be affected by climatic factor along with the dominant gene. Not only the genetic factor, climatic factors also affect. That's what Murthy said. And detection of apomixis. How we can detect whether it is apomixis embryo or amphimixis embryo? By cytological analysis of embryo cell development is a common used technique to study the apomixis. Aposporous embryo sacs are genetically 3-cell egg operators and central cell. No antipodal cells. By staining, we can easily observe it. Okay, and callose fluorescence in combination with the clearing of ovules can be used to detect the diplospory. It lacks in apomictic embryo sac, callose, which are present in the pollen tubes. Okay, and even in the style that is absent in the apomictic species. And uniformity of progenies from heterozygous or cross-pollinated parents is the best identification of apomixis. Uniformity. And In case of obligate apomixis, progeny from cross resemble female parents. Okay. In case of facultative apomixis, percent of progeny identical to female parent gives the degree of apomixis. In self-product generation, presence of plant showing no inbreeding depression is indicative of apomixis. Okay. We also can detect by molecular markers or Adventive, and also by embryological studies of embryoes confirm apomixis. And determination of apomixis, we can easily study the genetic study by using the recessive as a female as a recessive white one with the dominant homozygous. Now that we get absence of dominant phenotype in the progeny indicate the presence of apomixis. Here, dominant homozygous option and recessive types are present. By that, we can easily find out these are the apomixis species. And determination of amount of cross-pollination also we can identify. With the dominant homozygous, recessive traits in the mixed time allow the open pollination. By that, what happened? Here, dominant phenotype in the progeny of recessive plants. So, what happened? Here, proportion of dominant phenotypes in the progeny also present. There in the only recessive, here dominant also present. It indicates extent of cross-pollination. Okay? And applications. Main application of the apomixis. Apomixis is used to achieve following objectives that are apomictic plants resemble the parent plants almost all aspects. By this method, we can create more plants with the same genotype. Parental characters can be retained for many generations. Paternal inheritance. Now, it is a maternal inheritance will happen. Why? Because mother cells, from the mother cells only, it will generate. So, it retains the mother characters with the long generations. Fixation of heterosis. If the mother plant which we selected that is having the high heterosis that will follow the apomixis from the long generation. Rapid production of homozygous lines also impossible, and production of phenotypically stable population called hybrids also possible by using the apomixis. Okay, thank you very much, thank you all.